Hello student, this is Smita Mam working as an associate professor in Ravi College of Pharmacy. Today we are going to discuss about a pharmacognosy practical based on evaluation, specifically physical evaluation. Evaluation is confirmation of identity, purity, quality and detection of the presence of adulteration in a crude drug. Evaluation can be done by various methods, namely microscopic evaluation, which is done on the basis of the cellular structure of the drug, or mostly done for unorganized drugs. Macroscopic evaluation done on the basis of morphological structure of the drug. Then chemical evaluation done based on the color reaction on treatment with some specific reagents. Then physical evaluation, which mostly done for unorganized drug but can be done for organized drug as well. And the biological evaluation, which is based on the therapeutic action of the drug. Today we are going to discuss about the physical evaluation. Physical evaluation can be done by various methods, namely moisture content determination, ash value, extractive values, swelling index, foaming index, etc. Our objective today is to determine the foaming index of the given group drug. The drug we are using is liquidus. The foaming index is a measurement of how well a liquid extract can produce foam, primarily used in herbal medicines and natural product research. It indicates the presence of surface active compounds like saponins, which can be used in various pharmacological activities. A high foaming index suggests a high concentration of saponins in them. Saponins give persistent foam when shaken with water. Hence, plant material or extract containing saponins is evaluated by the measuring the foaming ability in terms of foaming index. Significance of the foaming index. It helps to assess the quality and consistency of the natural medicines. It can be used to screen the herbal extract for potential surfactants and active pharmaceutical ingredients. They have the ability to hemolyze the blood cells. Applications. Herbal medicines. It is used in herbal medicines. It can be used to identify plant with high saponin content, which have the expectorant, anti-inflammatory and antimicrobial properties. It can be used in cosmetic and pharmaceutical industry also to evaluate the foaming ability of the surfactants in formulations like shampoos and detergents. The factors which affect the foaming index are usually factors like concentration of saponins, method of extraction and type of plant material are the common factor which affect the foaming index. The drug which we are taking today is liquidus. Liquidus, as we see, the biological source is Glycosides agravia. The plant part used is dried fruit or stolons of the whole or cut, peeled or unpeeled drug. The plant belongs to the family Leguminosae. Chemical constituent present, the main chemical constituent present are triterpenoidal saponin glycosides. And uses, it is used as a flavoring agent, demulcent, mild expectorant, anti-gastric activity in peptic ulcer and many more. Now, when we talk about foaming index, so how to assess the foaming index? Usually, the foaming index is assessed based on the height of the foam formed in the extract. If the height of the foam in every tube, usually we use the test tubes. So, if the height of the foam in every tube is less than 1 centimeter, it means foaming index is less than 100. If the height of the foam in every tube is more than 1 centimeter, then the foaming index is more than 1000. In that case, where the foaming index is more than 1000, the practical is redone. Foaming index usually range from 100 to 1000. If the height of the foam is 1 centimeter in any tube, the volume of the plant material decoction in that tube, let it considered be A, is used for the determination of the foaming index using the following formula. Foaming index is usually 1000 divided by A. Remember, A is the volume of the plant material decoction in a tube which contains 1 centimeter length of foam. A is the volume in ml of the decoction in the test tube showing 1 centimeter of foam height. If the test tube showing 1 centimeter of foam height is the first or the second one, we usually prepare an intermediate dilution to obtain the precise result. For example, if A is 9, that is 9 ml of decoction is used, and then the test tube will be number 9. If A is 9, then the foaming index will be, as per the formula, 1000 divided by 9, that is 111.11, that is within the range of 100 to 1000. Another example, if A is 8, then the foaming index will be 125. Similarly, if A is 6, the foaming index will be 166.66. In all the cases, if we see, the foaming index ranges between 100 to 1000. And so, now let us do the practical.
Measure 100 ml of distilled water and boil it on heaty mantle. Now weigh 1 gram of powdered crude drug liquidus and add it to the boiling water. Stir and maintain the moderate boiling for 30 minutes. After 30 minutes, cool the extract and filter. Now collected the decoction or the filtrate in 100 ml conical flask and adjusted the volume of the extract to 100 ml using distilled water. Pour the decoction into 10 test tubes as 1 ml, 2 ml, 3 ml up to 10 ml and adjust the volume of the liquid in each test tube to 10 ml by adding sufficient quantity of water. Here we have used measuring cylinder directly to make up the volume. Close the test tube and shake them lengthwise for 15 seconds. Now we can see the formation of foam in all the test tubes. Allow the test tube to stand for 15 minutes and then measure the height of the foam in all the test tubes. The test tube containing 1 centimeter of height of foam is considered for calculating the foaming index. Thank you.